introductions now, so let's get this thing out of the way. Here you have your spear of battle. Okay, thank you everybody for coming today. For those of you that don't know me, my name is Zia. I'm the director of the Genomics Institute Office of Diversity Programs and the On-Demand Research Training Internship Program, which is an undergraduate diversity and research training program funded by the National Human Genome Research Institute. Today, I'm super excited to bring our speaker, Dr. Shaka Cunningham, who is a prominent research scientist in genomics. Sophie will be telling you more about his titles and roles, and he will be talking a little bit about his own path to his current position, as well as some of the initiatives that he's involved in. So I'm going to now turn it over to Sophie, who is the Genomics Institute Director of Diversity in a more general sense, and also one of our MCD faculty here. So Sophie?
Yes, great. Thank you. Yeah, so as Zia said, I'm a professor of MCD biology anyway, and a member of the Genomics Institute, and currently serving as the faculty director of diversity. And in that role, I get to lead the Genomics Institute Diversity, Equity, and Inclusion Committee, which comprises of faculty, technical, administrative staff, postdocs, graduate students, and undergraduates. And we are almost always recruiting for people, so when you see those emails, please consider joining. We do a number of things around promoting a more equitable culture in the Genomics Institute, and then also thinking about anti-racist policies and actions in the course of our research. And as part of that, we are really proud that we are actually we're going to be putting on a symposium on May 19th. It's going to be a half-day symposium on advancing anti-racist practices in genomics. And this is going to include workshops, panel discussions, and a keynote lecture. Please stay tuned for more information about that. And in order to help get us ready to really engage in this activity, we are trying to, in the lead up to that, have some seminars, more sort of traditional research seminars to get us there. And we're super proud to be partnering with Pathway to Genomics to have Shaka Cunningham here today. So as Zia mentioned, Shaka is Chief Scientific Officer of Polaris Genomics, which is a company that is focused on bringing precision medicine to the diagnosis and treatment of mental health disorders, including PTSD. So Shaka got his BA in biology from Princeton, and then received a PhD at the Rockefeller University in the field of HIV research, and then went on to postdoctoral work at the Institut Pasteur in France, or I assume in France, and NIH in the fields of immunology and tumor immunotherapy. And then it looks like your first real job was at Bristol Myers Squibb in development of cardiovascular drugs, and then he went on to several positions with the VA Health, and I think we're going to probably hear more about that in your talk. So I'll stop going on and on about you. But I think as we're going to hear, Shaka is really walking the walk and trying to really promote equity in both engagement and focus in genomics research. And so it's a real pleasure to have you here. Thank you.
Well, thank you so much. And it's a real pleasure to be here. Thank you. This is actually my first time on UC Santa Cruz campus, so I appreciate the hospitality and appreciate the invitation to come and speak to you. I couldn't think of a better way to kick off Black History Month than to talk about being anti-racist, particularly in our field in genomics and precision medicine. It's so important. It's a really vital topic that is of deep personal importance to me as a person of color who also happens to be a scientist. And it's something that impacts everything I do in my, not only in my professional life, but also in my personal life and the work that I try to do in the community to make it better for everybody. So I'm excited to talk to you today. I'm going to speak on the topic of engaging diverse communities in genomics research and precision medicine and share some of the work that we do in the community. I'll talk about a bit my personal journey with genomics and precision medicine, and then we can kind of wrap up and get more into the discussion that I was having with some of you over lunch about career trajectory and different ways that I've navigated a career in science so far. So let me get started. First, I want to give a disclaimer because I'm going to be speaking a little bit about the NIH All of Us program and some of the outreach work that my organization, the Faith-Based Genetic Research Institute, is doing in partnership with them. So I wanted to make sure you were aware of some of the terms that we use. And when I talk about engagement, outreach, and recruitment and retention, these are some of the definitions I wanted to make sure everyone was familiar with. But for me, when I think about my entire scientific journey and the times that we're living in, it's all about the DNA. So there was a popular 90s song by one of my favorite rappers, Puff Daddy, said it was all about the Benjamins. I said, no, it's all about the DNA, right? Deoxyribonucleic acid, this amazing molecule that makes us us, that as a molecular biologist, I never get tired of studying. And I really think that we're at an amazing inflection point in our understanding of DNA. We're literally in what's called the genomics era, the genome era. We're at the place where we're sequencing genetic information, producing genetic information faster than we can even analyze it. So that's a really, really cool place to be and a great time to be a student of this, because I can remember when I was a student many years ago, we weren't there yet. They hadn't even had companies like Illumina or 10x Genomics or any of these other PacBio big companies out there doing sequencing at scale. We were still in the early days of this stuff. I can remember having to print my own microarray, like dot my own thing on a glass slide to, oligos on a glass slide to try to get the kind of data that we get today in seconds. So it's very exciting to be in this area. Some other things that are exciting me about this is that as we think about this question of diversity and why we should do it, some basic facts about humans and how we evolve bring the cause of diversity to the fore. The fact that we all as a species originated in sub-Saharan Africa. That is a scientific fact that is pretty much undisputed, that early humans evolved over 100,000 years ago and migrated out of the continent of Africa. So understanding diverse genomes, particularly African genomes, becomes very important. And what I've learned at several different research conferences that I've attended recently, that our understanding of African genomes is very minimal at this point in time. That is that less than 2% of all of the genomes that have ever been studied are from people of African origin. Now, you might say that's bad for people of African origin, but given that we all spawned out of the continent of Africa, that's bad for all of us, because that means that we have less understanding of the total diversity of genomes that are out there. So I think that diversity from that perspective is really important. Some other facts that I like to reference when I think about diversity is that as diverse as we all are, we as humans genetically are actually remarkably similar, right? So if you look at the NIH website, I pulled these numbers off the NIH website, is that our genomes are more than 99.9% identical. Then depending on who you talk to, that number can shift a bit, 99.9, 99.6, but the bottom line is remarkable homology within our species. When you think about primates, our genome is roughly 98% homologous to chimpanzee genome. So we are the same species, and that 1.1% genomic differences are reflecting all of the diversity of phenotypes that we see out here. I think that's amazing to me, because I think that tells me several things. One, I really want to understand how all of this diversity is captured in that 0.1%, but then I also really want to appreciate how we're all part of the same species group, and how this whole concept of race as a thing has no real biological relevance, in my opinion. We are biologically one race. It's called human, and many different genotypes within it. So that's helped me kind of change my view of race from as a biological thing. I don't really believe it as much. It's more of a social construct. That's how I view race and how we've experienced it. And as accordingly, for some of those reasons, a lot of the studies have been biased in certain to certain groups, particularly those of European ancestry, making those from other ancestries tremendously underrepresented. And this has caused a tremendous problem in the field of genomics that the NIH and other agencies and the medical community writ large is trying to correct. They realize that diversity in genomics is good for all of us. And so how do we really implement this and why? So some other reasons on why is that your generation and the generation right below you in this country is growing, and the diversity of that is growing. So right now, under the age of 16 in the United States, we are a majority minority country right now. So if you look at all of the kids under the age of 16 and you have said what racial ethnic groups do they belong to, it's no longer majority Caucasian. And so I think research, particularly genomic research, has to keep pace with this demographic change that's happening in our country, that'll happen in your lifetimes and particularly in my kids' lifetimes. Also, the other thing that's been happening that's amazing, that's really driven this technological advance, is the capacity to sequence things at scale. This is a graph that's also from the NIH website that shows the progress we've made since the first draft of the human genome was issued back in 2001 until 2019, where the cost per human genome has dropped precipitously as the sequencing technology has improved. It's dropped faster than Moore's law, which is the amount of information you can cram onto a microchip. So from a technical perspective, the genomics industry is killing it versus the tech industry in terms of its ability to generate data at scale. And that should make you even more excited to go into this field and make your presence felt. Literally, as the cost of sequencing goes down, as it's done since the first human genome, to now we're at about the $100 or $200 whole genome. If Illumina wanted to tomorrow, they could offer a $100 whole genome. They just announced a new sequencer that's a really amazing instrument called NovaSeq X that can sequence genomes, whole human genomes, 30x faster than ever before. And that can allow us to lower the cost, and that will democratize access, which is really going to drive more data and more research and more understanding of the genomics of human disease. And that's what programs like the NIH All of Us program are trying to do. So you have many efforts. Our government, I'm really privileged that we live in this country that really values research and development. And I, as one who did a postdoc at the NIH and actually worked there for several years, got to appreciate the importance of government sponsorship of science. And one of the things I'm proud of that our government did sponsor are these big, what I call population genomic studies. And so the NIH All of Us program is one of those studies, which is goal is to move us from the current paradigm of the sort of one-size-fits-all medicine, where they give you a drug at a specific dose, no matter what your genetics are, to a sort of more informed medicine, stratified medicine, where certain attributes about you are considered, to precision medicine, where very specific attributes, like your genomics, other things like your health history, other factors like your lifestyle and other sort of social determinants of health are also considered, that will really create a profile to understand not only what medication you should take, which dose of that medication you should take, and how effective it's going to be for you. Going from that one-size-fits-all to precision medicine is where this entire research effort is moving. And that's why they founded the NIH All of Us program. So I was very proud to be working in the government. I was actually at the Department of Veterans Affairs when President Obama launched the Precision Medicine Initiative, which was this grand idea to sequence millions of people. So you had kind of two arms of that in the government. One, which has become the NIH All of Us program, which the NIH is coordinating. It's going to sequence 1 million citizens. The other was housed within the Department of Veterans Affairs, called the Million Veteran Program, which was sequencing 1 million veterans. And as part of the VA during my time there, I got to work with the folks who are orchestrating that project and be a part of that project. And that was a very, very exciting opportunity. What I learned, though, is that there's still a lot of persistent bias throughout our system with regard to precision medicine and genomics research in general. And this is actually from a paper that was published back in 2009 and then follow-up studies in 2016, when you see this tremendous increase in the number of samples studied, genomic samples. Back in 2009, about 1.7 million samples. 96% of those samples at the time are of European ancestry. Now, we know the world is not 96% of European ancestry. It's really more like 15 to 17% European ancestry across the globe, okay? So you actually should almost invert that number to be accurate with the globe. Folks started to realize this. Other studies started happening in Asia and other countries, India and China. And so by the time about 7 years later, you had 30 times more samples. And still, though, this big disparity as far as European ancestry dominated. Now, the needle hasn't moved much. And if you notice this little sliver down here, other non-European, including African American and Latinx and others, that's the sliver that we're still kind of looking at. And that sliver has to grow. And it hasn't grown proportionally. Remember, I told you, less than 2% of all the genome studies have come from African origin. So we still have a lot of work to do. And even though the NIH wants all of us to be for all of us, we're going to have to work hard to make it for all of us. And that's where engaging communities becomes so important. So I wanted to talk a little bit about the work that I was able to do on the Million Veteran Program, because that really informed a lot of the work I do in the community. That was an amazing program, and it's still an amazing program that began in about 2008 with a survey. And this is something that I think the VA did very well. They actually surveyed veterans on their attitudes towards genomics and precision medicine. And they asked a group of about 3,000 veterans, what is your attitude towards doing this? If we did it for free, if we'd offered you free genetic testing, would you do it? And they ranked those responses by group and by gender and ethnicity. And it was some amazing sort of responses that we got. But this program has been tremendously successful in recruiting veterans. Now over 800,000 veteran samples have been recruited. And I'm happy to report that the diversity goals that we set out for that program were actually exceeded. And I'll show you how we did that. So this was actually the data from the study that was released back in 2009 of veteran attitudes regarding genomic medicine. And when you asked white non-Hispanics, if we offered it to you for free, would you participate? 74% of them said yes. If you asked Hispanic veterans, which was about 7% of the veteran population, 82% of those individuals said yes, we would participate. We trust the VA enough to do it. All others, non-African American, Asian, Native Americans, Asian Pacific Islanders, about 71%. But African Americans were way down there at 51%. And I can remember being in the room when they unveiled that data. And I was one of 2 black people in the room, and I was the only scientist in the room. And my bosses at the VA looked at each other and said, they didn't even look at me. They looked at each other and was like, why don't black veterans want to do it? And I was like, I know. I'll talk to you guys after. And so thus began a plan to actually address this issue. And my advice to them, which I'm glad that they followed, was rather than sort of sweep this fact under the rug, let's attack it head on and use it as a teachable moment and an opportunity to actually get in front of this and change the dynamic and change the narrative. And we did that. And I'm very proud of that. And so what that survey showed us that African Americans were very much less likely than their Caucasian and other minority veteran peers to participate. So what they allowed me to do was create and set up what we call the Genomic Medicine Diversity Working Group. Okay, they had an advisory committee for the precision medicine program, and so they allowed me to create a subcommittee of that, where for a year I gathered experts in the field of genomics research who were people of color, who are familiar with the communities, who have worked in the communities, to help us design a strategy to recruit and actively recruit minority, particularly African American veterans. And so we came up with a set of strategies. Some of the lessons that we learned was that you need to take some extra steps to reach out to communities that are less likely to participate, to help to restore trust, which was often the barrier for minority veterans to participate, to encourage them to participate. Then some other common sense things, like oversample in areas where you had high African American veteran population. So if you know that African Americans and others, veterans are in major cities, in big VAs in major cities, really make sure you have strong recruitment at those sites. And then really work hard to build the trust by creating ambassadors for the program that were from these veteran populations. So working with African American veteran service organizations and other groups that dealt with African American veterans to share information about the program was a very effective strategy. And as a result, even though African American veterans are only 12% of the overall VA population, they're about 16% of that data set. So that's amazing. Out of the 800,000, 16% of that's African American. That's by far one of the largest African American genomic cohorts available. Okay, very proud of the work. What are we fighting against, and why is it so hard? So this friendly looking gentleman here is kind of part of the genesis of some of the history of that we're fighting against. This sort of persistent, long history of racial bias from within the medical community. This is Dr. Samuel Cartwright. He was a prominent physician back in the 1850s. And he actually wrote a scientific paper called A Report on the Diseases and Physical Peculiarities of the Negro Race. And this and documents like it were the sort of genesis of this movement that was in the U.S. at the time, called eugenics and phrenology, these sort of pseudoscience sort of fields that really gave birth to the same kind of propaganda and pseudoscience that Hitler's Nazi Germany used against the Jews. But in this case, they would come up with all kinds of random conditions and attribute them genetically, and say that they were differences between the races. So, for example, he invented a term called drapetomania, and he called it was a condition that was a genetic defect in enslaved Africans that made them want to seek their freedom. So can you imagine that? Someone wanting to seek their freedom when they're a slave was, according to him, a genetic defect. So this was the medical establishment of the time. So that only grew and sort of, festered in the country for the next 100 years. So you fast forward there, and you lead to things like the Tuskegee Public Health Service syphilis study at Tuskegee University that was down in Tuskegee, Alabama, that was from 1932 to 1972. It was a 40-year study that the U.S. government actually sanctioned, where people were purposely injected with syphilis when they were told it was not that. They were told it was a cure for this stuff. So that has sort of left a psychological scar on many an African American. That is kind of exhibit A in some of the distrust more recently. Then Henrietta Lacks, which many of you have heard about, literally had her body taken from her without her permission and used to actually, her cells used to make billions of dollars worth of medical discoveries throughout the decades. And then I would say exhibit C, which is that for African Americans, is that sort of disparity in their healthcare experience that many folks complain about from not receiving the same standards of care. So when you think about it from that perspective, you can sort of understand that distrust number that we saw for the African American veterans, and that same kind of distrust number we often see when we poll African Americans in the broader community. So how do we kind of overcome that? Well, some of the strategies that we engage with in the community is to highlight the health benefits that minority patients can see due to advances in biomedical research. One of the strategies that we do through the Faith-Based Genetic Research Institute, my nonprofit, is to provide culturally congruent, honest brokers. That is, people who are from the community talking about science to the community. So folks like myself and my colleagues who are researchers and clinicians in the field, sharing information. And then I think it's incumbent, the last point is incumbent upon all of us to share information that makes racism sort of not welcome in places of higher academia, in industry and government, to truly create a level playing field. So this concept of being anti-racist is not just for the people of color, it's for everybody, okay? So, I mean, if you look at what's happened recently in our society, you saw in Memphis, Tennessee, 5 black police officers beat a black innocent person to death. And racial bias doesn't matter where it's coming from, the race of the individual instituting it. So we want to stamp it out every place that we get it. Some examples of some events that we do in the community that are very helpful. We do lectures like DNA 101, which educates the community. This is my colleague, Dr. Kareem Washington, who used to be chair of genetics and human genetics at Howard University, a historically black college in Washington, D.C. We actually go into the communities and talk about genetics and health. And we talk a lot about genealogy and heritage, because we found that particularly African American populations really want to understand that. And that makes sense when you think that the average African American family can't trace their family history back past the 1800s, because slave records weren't always well kept. So if your parents were brought here, if your ancestors, like some of my ancestors, were brought here as enslaved individuals and their records weren't kept, you lost track of them. So there's a real disconnect and a desire to connect with ancestry in Africa. And then I wanted to kind of shift gears and talk about why I'm not so much not only enthusiastic about genomics, but why it's personal for me. And I've always viewed your genetics as one of the most personal attributes about yourself. And by show of hands, how many of you have been sequenced? Even over the counter, 23andMe or Ancestry or anything? Okay, hands down. How many have had a whole genome sequence done on you? Wow, army of one. Okay, 30x whole genome. I've done it myself too. And I've had it done. Highly recommend it. And there are services that allow you to do that. Check online. And you'll learn some fun stuff about yourself. My genome, I'll just share with you, I'll even show you a little bit of it, is not really that exciting. I don't have a lot of cool mutations. I have one that gives me fast twitch muscle tendency, which kind of explains why I used to be kind of fast when I was a kid, I think. And my kids are pretty fast. I think I passed that on to them. And then I have another that makes me process certain drugs differently. It's actually a cytochrome P450 variant. It makes me process certain antidepressant drugs, which if I ever had to take an antidepressant, it'd be helpful for me to know. So, but that, to me, gaining that knowledge, the reason why I really wanted to do it was to understand more about myself. Now, this is me back as I was being formed. These are my parents. You might think they were in an African village, but they were actually in Washington, D.C., in a place called Malcolm X Park on their wedding day. And this is me in my mom's belly. I call my mom an African child bride. She was actually 20 years old, but she looks very young. And my dad with his big dashiki and huge afro back in the 70s. This was me, and this is my genetics came from them, okay, which they got from their parents, but wanting to understand them. And by virtue of my grandmother, who gave my mom her genetics, she really inspired me in science. This was my hidden figure. My grandmother, Alfreda Simmons, who passed away, was a cancer researcher at the National Cancer Institute. And she gave me my start in science when I was 8 years old. So when she first put a pipette in my hand and showed me how to do PCR, that was when I became hooked. This is us in visiting an actual slave fortress in Dakar, Senegal, when my mom was there doing a Fulbright scholarship in fine arts. So this is me at 8 years old, touring a slave fortress for the first time in my life. And I can't tell you how viscerally powerful that is when you're an African American who you had ancestors who may have passed through that slave fortress as a child, touching those chains. I still feel it 40 years later, right? And, my grandmother was not only there guiding me through that, but she was my inspiration in science, kind of like a hidden figure, like you watch that movie about the hidden figures in NASA, like Katherine Johnson and her colleagues. My grandmother was like one of those folks, but inside the NIH and NCI. And, as a result, I still love working in the lab and talking about science today. She really inspired me to do what I'm doing. But my real personal why for understanding my DNA is for my kids. And these are my two sons, Logan and Sam. This is us at the baseball game a few years ago. I want to understand all of the variants in my DNA that I'm passing on to them, good and bad, so that I can give them a leg up health-wise and help them be able to get in front of any issues they might have. Now, like I said, luckily, I don't have a lot of mutations that seem to be bad. So I'm kind of, I think good. And so in addition to the fun kind of ancestry stuff I learned, I wasn't quite as African as I thought I was. I had 3 African names, but I only have about 56% African DNA, right? So a lot, and that's many African Americans have racial admixture from Caucasian and other sources of DNA, but that's what makes us, super interesting genetically. And all the more reason we have to understand everyone's genetics. And so I really want to understand that. My personal reason is for my kids. And it is good science. I mean, so part of the reason that drives me to really get in the community and make sure that we have more diversity represented, I want the genomic databases that are being used to develop the medical tests of the future to be broadly applicable to all communities. It's good science. And there's many studies. This is one from Nature showing how important it is to diversify our genomic data sets. And it's something that we really need to do. So I'm encouraging us to all be steadfast and do this work, get out there, engage with diverse communities, listen to the communities that we're seeking to serve, and that everyone, regardless of your racial or ethnic background, just be an advocate for each and everyone's group. And finally, I'll leave you, if you did want to participate in all of us, get your genome sequence. It's free. It's actually a free service. You're over the age of 18. You can hit this QR code, go to it, sign up. I did it. It's actually pretty cool. For some of them, I think they even give you a Fitbit or something like that. I didn't sign up for that part. But there's all kinds of information that will be gained for that, and I highly encourage folks who are interested to check that out. And that's where I'll stop now and then talk a little bit about my career. So I'm going to stop my kind of formal presentation there and get into my career. But before I do that, any questions about what I just said? All right. One in the back. So that's a great question. One of the advantages of sequencing veterans is that the VA, which was really cool, was one of the first, I'd say, hospital systems to have an electronic health record. So they had been keeping electronic health records, monitoring veterans' health attributes for over 40 years. So they probably have the deepest repository of longitudinal clinical medical data in an electronic health record. So when you match that data with the genetic data, it's a much easier path to making associations that help you understand the genetic basis of diseases. And a lot of good work is starting to come out of that project as more and more of those those samples are sequenced. Any other questions? That was a good one.
It's amazing that you ended up enrichment of African American DNA in the Human Genomes Project. And while there's no short answer to this question, how did you do that?
A lot of hard work. It took us several years. I actually formed a committee of experts, and we met for about almost about 18 months straight to plan our strategy, review it with the project leads on that program, get it endorsed by the leadership inside the VA, and get it implemented during the recruiting strategy. And to the VA's credit, they really followed through. They took it seriously, and they did it. And I'm hoping for the same from the NIH All of Us program, of the Citizen program. It's a little bit harder, I think, for all of us because they don't have a consolidated system of hospitals to recruit from. They're kind of recruiting it all over. But I think that with a heavy focus on really meaning that they want to do diversity and really showing the community they want to do it, I think they will have similar success. And that's why FBGRI, we're happy to work with them.
Diversity, we really do want to sample as broadly African populations as we can, right? The more that we discover from many traits in our specific African population, and some of the European specific. So how do you go? Obviously, the slave trade was biased. The slave trade was biased. So how do you go about
So you need. Well, the NIH has some efforts that were funded years ago with the Wellcome Trust. They had something called the H3 Africa Initiative that was collecting cohorts from different parts, mostly of Western Africa. But what's missing is, you're absolutely right, is a kind of a holistic view of the continent. I think what we need to do is not just the NIH. I think it's governments. I think it's corporate entities. I think it's nonprofit foundations. is fund more research and sequencing capacity on the continent of Africa to enable that activity to happen where people are, right? Because we're not asking London or England, Genomics UK, to sequence us, right? We're doing that here in the US. We have capacity. So we need to have all countries actually generate that capacity. And I think companies like Illumina and others are trying their best to get there. But I think that is a collective effort of government, nonprofit organizations, as well as for-profit companies to really fill this gap of a lack of genomic knowledge.
South Africa in COVID showed us they have capacity.
They do. But are we sequencing genomes at scale? Are we doing these sort of population genomic studies with those samples? Maybe not, right? Because the COVID protocol is way different than a 30x whole genome sequence. Yeah.
With respect to the entire human population to have their genome sequence, how do you address the issue with being anonymous or having your metadata associated with your clinical data? How do you move towards, if everyone's going to be sequenced, how do you keep yourself anonymous with that data accessible and available to researchers around the world?
I think that's going to be a big challenge. And I think the people that figure out data security with this are going to make a lot of money, right? Because you're not going to really stop the train. The train is moving. But I think the best way will be, just like you do with bank information, find better ways to secure it. Okay. Now, I do also believe, and this is my personal belief, so don't quote me on it, or you can quote me on it. I think that some of the fear is a bit irrational, because I often tell my students, when you look at a DNA sequence, it's a bunch of A, T, C's, and G's. If I don't know really much more about you, there's not much I can do to hurt you with that, right? Like if I know that you have a mutation in your HLA-B57 that makes you abacavir hypersensitive, then maybe, yeah, I could do you some harm by giving you a drug that I know that'll give you an anaphylactic shock type reaction. But short of that, and that's a very rare mutation, not much more I can do to harm you with the information. You see? The only thing that you could say that could potentially do harm is if you identified somebody. And but in the cases that you've seen publicized in the media, they're identifying people that were actual criminals, right? That had actually done some pretty heinous things. And I don't know about you, I'm okay with that. Like if it was a relative of mine and my DNA helped catch somebody to kill somebody, take them to jail. But that's just, again, that's my opinion. So as far as weaponizing the genome, I don't know how much that's really possible. But I do think we still will need, for data security purposes and to make people feel comfortable, find better and better ways to keep that information secure.
So I agree completely. Just the ethical dilemma, right? That you're supposed to have action with, or that permits people to have self-determination and freedom. And so by me going to get my genome sequence, I've kind of almost forced my parents to get some portion of their genome sequence, and because they haven't explicitly consented to that, there's an issue with consent, and I forgot what the word is off the top of my head, but that idea that they should be able to make that decision for themselves.
Yeah, they should. And this kind of idea of cascade testing of families. I'm a part of a project called BabySeq. I'm actually a consultant on it. It's an NIH-funded project where they're going to be sequencing about 1,000 babies. And one of my colleagues is the PI on that project. And my role in that project is to make sure we got diverse babies in there, right? So some of the same kind of strategies we use to engage communities, we want to make sure we're engaging families. What's really amazing, some of the studies that my colleagues group has done at MIT Broad and Brigham and Women's at Harvard, is that they've done several kind of population genetic studies and even sequencing of objectively healthy people. That is, people who elected to get sequencing even though they didn't have any known health problems. And in cohorts of hundreds of people, they found about 8 to 12% found that they had a variant of disease-causing significance that they didn't know about. They were just walking around with it, but they had never been sequenced. Again, making the argument for getting sequenced. And then another 80% carried a variant that had some sort of clinical actionable trait, right? So that's actually really higher than I would have imagined out of a group, that 80% of us are walking around with some variant that may have pharmacogenomic or other sort of treatment implications for us if we were given a certain drug. So I think all that to say is that we definitely want to secure this information You definitely want to preserve individuals' rights. But I've seen numerous cases from studies like those, like, for example, the Baby Seek project, where an infant had a mutation that showed the parent had a risk of the disease. The parent elected to go and get sequenced, caught that risk, confirmed it, and was able to take action to save their life. And in one case, it was like a situation of a severe breast cancer that a mom was able to go and address that so that she's now alive to raise her child, right? That's a serious kind of situation, which wouldn't have been enabled without the child being sequenced. So you offer the parents the choice, and they always have the choice not to do it, but you also want to offer parents the choice to do it when it makes sense.
Curious about like the economics that you're talking about. So you're talking about how the decreased price of genome sequencing has gone down from what billions to dollars today. Has that led to increased availability and lower cost allowed developing countries to do this themselves? Because you're talking about how you mentioned how big part of it is just Caucasian. Has the decreased price allowed developing countries to do that for themselves and come up with their own?
Well, that's the hope. And I think not all developing countries are like so resource poor, right? You have some countries in Africa, like Nigeria has a ton of money from oil revenue, and other. South Africa has a lot of resources from natural resources and other things. So it's about the investments, though, that the countries themselves make. I think other countries in Africa, like, Eastern African countries and others, and even some countries who are struggling more with war and conflict may not be in the right position to do it. But the hope is we can get there. So that's why I believe that NGOs have a big role to play in this. And I think that the companies themselves have a role to play in democratizing access to the companies. Because if they really want to empower the sort of genomics era and make it applicable to everybody and make it the best it can be, they've got to empower studying diverse genomes. And I feel like they are talking that way, at least at the conferences I've been through. But the proof will be in the pudding to see if that activity actually happens. Any other questions?
Get into sort of giving back. So you take somebody's sample and you sequence it. What do they get back out of it? And how do you frame it in a way that is meaningful?
Well, that's a good question, because a lot of the time, in addition to your personal health information, like, again, when I participate in genetic studies and learn something about myself, that's knowledge that I now have and I can pass on, right? And so that's something I know that I didn't know before, and that could impact a particular health action I take if I need to take it. For example, I told you my cytochrome mutation, that if I took a certain antidepressant, I want to know that. So letting them know the benefit there of them knowing. And then I think,
So, like in the veterans study, would they get a report back?
yeah, so that study wasn't designed for return of results, but I know they've amended it that if there was an actionable mutation, they had certain sort of services and other things made available to them. So if it was something sort of popped up that was, on the radar, then, they had the opportunity to kind of circle back with their provider and go deeper with the sequencing, et cetera. Um, so, but the NIH study, for example, does return some of your results, and it does tell you some things about yourself, and it's pretty cool. And they kind of update it as more knowledge is gained about your specific mutations. So I think that's actually a good attribute. And then I think, again, the whole benefit to the group of understanding our collective genetics to help us better design new treatments and therapies. So we're finding new mutations every day that underscore different disease conditions. And now that we're in the era that we can edit genomes and do things to actually fix things, we have to know what to fix first in order to be able to fix it. So I think that's one other reason to really lean into genomics and participate.
I have a question. So, I know in a lot of projects, the government tries to talk about like having the private sector take over for some of that stuff. Why is the
NIH.
thing? Yeah. Why are they taking such a big step? Aside from the diversity, like are they patenting like genes or something? Or
No. So there's been some very interesting, and this is a good career. How many of you guys ever thought about law as a career path? right? So that's a very lucrative field in this patent law around this. And I tell you, as someone who just shelled out a bunch of money to a patent attorney to file a patent for my company, um, the it's a very interesting and evolving field. You can't really patent a gene because it's a thing of nature, right? But you can patent an algorithm or a way of measuring different attributes or markers and designing that into an algorithm that can predict X, Y, or Z or condition or define things. So that's what a lot of companies have done. But the validity of any patent that was based on a gene, I think, is called into question by this Prometheus ruling that happened several years ago. And so I think, that space is going to be a lot of case law for the next several years, figuring out, where that's going to land. But I think what's really exciting is not necessarily being able to patent a gene. It's to come up with a new way of defining conditions. And I think when you look at areas like cancer, which is the furthest ahead in precision medicine application, one of the attributes of cancer that made it kind of ideal for that is that you kind of know what cancer looks like. You kind of know what a breast cancer cell looks like versus a prostate cancer cell. There's a lot of pathology and data around that defines what those conditions are. But if you look at another area like mental health, a lot of more gray area. What's the real difference between PTSD and depression molecularly? We don't really know that yet. What's the difference between depression and bipolar disorder molecularly? We don't fully appreciate that yet because a lot of the symptoms overlap, but the actual phenotype of the disease could be different. And that's what the research that my company and many other academics out there are doing to try to design that. But once we know it, imagine how much better we're going to be able to treat it. Just being able to distinguish between those two conditions will completely change the treatment paradigm and improve health outcomes right away. And that's something that really drives us to the work that we're doing at Polaris.
So, do you feel like you're making a move away from genomics and now into the multi-omic space with like your diagnostic tools or studying PTSD and depression?
I love that term you use, multi-omics, because I think that's the future of it all. And I was talking about earlier about the central dogma, like DNA, RNA, protein. If you understand DNA, you only understand one part of the story. RNA, another part. Protein, another part. If you understand the entire continuum, now you're starting to have some rich insights. And so I'm a big fan of multi-omic research. In fact, we're looking at various tools from DNA methylation all the way to RNA transcriptomics to proteomics, the actual continuum in certain of our patient cohort data sets to really not only validate our biomarkers, but to give us more confidence and knowledge about what is the biology of the conditions, because that's what we're trying to really get at.
I think that's a good segue. While we still have time, I
Yeah.
wanted to give you some space to talk about your personal path and how you got to what you do.
Okay, great. And this is the fun part. So I was going to say that. So for me, this has been a long journey, and I got to kind of go down memory lane with some of you all at lunch. Thank you so much for that. Um, I sat in your shoes 20 years ago. Now it's hard to imagine that it's been that long, but it goes by fast. And I remember what it was like being an undergraduate and not really knowing exactly what I wanted to do, but having a general idea that I wanted to do something in science. Now, I was joking earlier, I was pre-med just like everybody else was in my class at Princeton. If you were a mobile major during that time at Princeton, like 99% of you were pre-med. But it took me doing an actual internship with a Princeton alum who was an MD and PhD who actually did both to let me know that I didn't really want to be an MD, right? I went to a hospital, first sight of blood, I almost fainted. I was like, I don't really want to do this, man, not every day. This is not what I want to do. But when he was an orthopedic surgeon, so when he was kind of reattaching a guy's finger, it made me almost faint. But when he was doing bone regeneration research, like drilling holes in rat bones, I did that all night long. I would stay up like hours, like putting little discs of material in the rat bones and hoping it grew and being very excited to see if they were walking the next day. So that let me know I really liked research more so than the patient care, and that really helped me understand the difference between an MD and a PhD career path. And it set me up. And then I've actually had a very enjoyable career So far, I've had a lot of really amazing mentors throughout my time from undergraduate all the way through graduate school, through postdoctoral, then into government and then beyond. And I say as a general rule, I try to collect mentors, people that you can learn from who are senior to you. Everyone that has progressed in their career that's senior to you can teach you something. So forming relationships with folks, having a few set of very key trusted advisors that I can always turn to when I'm making key career decisions, those are things that have helped me progress. Now, I've had kind of a winding path career because I started out as a molecular biology major. Then I went straight into pharma, which was very rare. You don't see that much. But I had a lot of lab research experience, but still not a whole ton of experience to start in pharma. So I was kind of lucky to get my first pharma job. I told the story at lunch. I sent out 100 resumes when I graduated. And I just knew that I was going to get a hit rate. Those of you who weren't at lunch, how many of those responses do you think I got off of 100 resumes?
At least
20? Man, I would have felt good. I got 3. And 2 of them, when I interviewed, they were like, you don't have enough experience, so it wasn't really a good interview, right? So one of them resulted in my pharma job. They gave me a chance. They were like, look, you don't really have a lot of experience, but we can tell you know how to do a bunch of stuff, and we can tell you really want to work hard. We can sense your passion. So they gave me a chance, and that allowed me to have my first pharma industry job, which I really, I was thriving in, really learned how drugs are developed from concept to actual clinical application, and got sort of a mini PhD before going back to get my PhD at Rockefeller after that. And then at Rockefeller, doing my PhD was the best thing I ever did. I highly recommend doing a PhD if you're passionate about science, because it was literally the best time of my life, just doing science all day, all night. I told folks earlier I used to sleep on the futon in the lab because I didn't really want to go home. I wanted to see how my experiments turned out. So it wasn't like a job for me because I really liked doing it so much. So if you have that kind of passion for it, definitely go into science and just play it out. And then, I was lucky. I got to have my choice of postdocs. I did pretty well. I graduated top of my class at Rockefeller. I was a valedictorian for them, I guess. And that was the first time I'd ever been valedictorian. It was a very kind of historic moment for me because I had my mom and my grandmother at my valedictorian speech, and I got to acknowledge them in front of this crowd of all these esteemed scientists and Nobel laureates that were at Rockefeller and these alumni, and let them know that's how I got here, and I'm here because of them. It was a very special moment for me to share with my family, probably the best academic moment I had, and then it kind of set me up for all of the remaining success I had as a postdoc, and then going on into government, and then into my own company. So it's been like a series of successive steps where I've been acquiring different skills to help me be better prepared for what I'm doing now. And that would be my advice to all of you, is to continue on a path where you acquire skills that are going to make you in demand within the marketplace, whether it's in a research environment or whether it's in an industry environment or a government environment. Those who have skills and knowledge will always be at a premium, and you'll be able to get jobs. So I highly recommend that.
A little bit about the difference in working in pharma versus NIH versus what you're doing now or doing academic research.
Yeah, they're very different jobs. I mean, it's all part of an ecosystem, right? Because the government funds research, right? So pharma companies are like vultures around data, right? So academics want to generate data because they're on the discovery path. They want to understand how things work. So they publish papers, they do studies, and they make discoveries in the lab. Pharma is looking at which of those discoveries can we apply to treat a disease. So what's a big market disease? So pick a disease. Let's say one of the diseases I worked on, heart disease, atherosclerotic heart disease. So what do we know about atherosclerotic heart disease? So look at all of the major academics that study that disease. What receptors are involved? What cell types are involved? How does the process of plaque development happen? How can we potentially stop it? And then they'll say to people like me who work in industry, go design a test to make me a drug that will do that, right? And so that's the kind of work I did in industry. I would develop assays to model disease conditions that we wanted to develop drugs for, right? And then once you develop a drug that has an effect, you gotta hope that when you put it into a body, it doesn't kill a person and it stays around long enough to have the effect. So it's got good PK and good pharmacodynamic and low toxicity and very few adverse events. And then if you're lucky, 1 out of every 10 drugs that make it sort of to late clinical trials will actually make it out to a drug that you see put in a person. And then when you look at the commercials, what do you hear on that? All the side effects, right? 50 million, because they're required by law to tell you every single side effect, which makes sense when you think about what you're trying to do. You're literally trying to pluck a needle out of the haystack, find a chemical compound that does exactly what you want it to do and nothing else. That's almost like magic. And every now and then you get lucky and you have one that does that with doing very little else, but most of the time you have a lot of other things you do. So pharma is different in that regard. It's really, its job is to commercialize the discoveries. And government is another job. It's kind of like steering the Titanic. Its job is to create the ecosystem for research to happen. So let's put that in context. If our government did not fund virology research, how would we have done for a COVID vaccine? Wouldn't have had it, right? Because even the researchers that were trying for decades to get funding for their research from mRNA and all these other things, and eventually kind of getting some funding, they wouldn't have even had it. We wouldn't have known what a coronavirus was. We wouldn't have the assays to actually test for them. So we wouldn't have been able to make the progress we did on vaccines and how fast we did it. So that's kind of how the whole ecosystems coexist, and they all kind of are meant to work together. And when things like COVID happen, sometimes they do work together very well. I think we should be very proud of the effort that our country did within a year to make a vaccine for a virus that had only been recognized maybe a year and a half earlier. That is amazing in human history. And so that is all due to the funding decades before at NIAID and individuals like Dr. Fauci and his colleagues, like really committing resources, time, and effort from a government perspective. So, yeah, and their jobs all throughout that ecosystem. This is what I want to impress upon all of you all. The key thing is skills, right? I think for all of you, develop more skills, enhance your knowledge, and become a thought leader. Those of you who go on to really hone your skills, learn how to do a lot of different things and apply a lot of different methods and become a thought leader, those will be the ones who will have the most sort of job opportunities, I'd say, in the future. Because we are in a knowledge economy. And, you know, there's a lot of competition for skills. And that's something that I would highly recommend that you continue to build as you continue your education.
We're coming up to the top of the hour. Any last questions? Last chance. Thank you very much.
Thank you all.
Now, I have a little something for you because I heard you talking about how much you like DNA.
Yeah.
So we have an event on our campus that some of you know about called DNA Day. And these are the official T-shirts of DNA Day.
Awesome.
I'm doing this as a plug too,
Right.
because I want people to turn out for DNA Day.
That's all right.
There are 4 of them.
ATC and G.
You got it. And we do a dance called the double human helix,
Okay.
where folks form a double conga line. And
Okay.
they match up with their correct nucleotides.
Okay, T, where's my A? All right.